about the chromosomal theory of inheritance. So far, we've been talking about Mendel and his genetic ideas. And because we know about genes and we know about DNA, we've been sort of bringing that in and out as we've talked about it. But remember that Mendel did not know anything about that. He had no knowledge, no one did at that time, had any knowledge about, well, to be honest, even the knowledge of being able to see inside cells with such clarity as we can today. Back then, microscopes were not as good. We couldn't see DNA. We didn't know any of that actually existed. So what Mendel said, remember he used the word factors. And of course, he also coined his law of independent assortment, which means he understood that somehow, independent with an A, assortment, he also understood that these traits seem to be able to sort themselves out without being connected to each other. We know that's not entirely true anymore. And that's because we know that each trait doesn't exist on its own. So, he thought that each trait was sort of isolated on its own and existed and was able to be moved from mother to daughter to son and so on. But this is not the case anymore, because we know that the new idea, the idea of the chromosome, once it was discovered as the mechanism for the movement of genes. Remember, what we have is a chromosome. Here it is in its doubled copied form, ready for meiosis or mitosis. And we have these spots. And there are spots all over here. We'll draw a couple just for fun. Regions on the chromosome. And these, of course, are the what we call the genes. These are the alleles for genes. And we know that what a gene is now. The gene, of course, is a sequence of bases. Remember those bases, C, G, T, and A on the DNA molecule. So we now understand what that means. But you'll notice that the three different alleles that I've drawn here, the red, the blue, and the green alleles, they are on the same chromosome. One chromosome can contain many, many, many genes. And so the only way that genes can move around is by riding on their chromosomes and being assorted through the process of meiosis. So that means that we can break apart these two copies. Or, I guess what we could do is in meiosis, we could have the homologous chromosome that went with this, and we can pull those apart. But there's no way of breaking apart an actual chromosome. So whatever genes are on this chromosome all move together in the process of meiosis. So that means that these three genes, they are linked together chromosomally, if that's even a word. They move together on the same chromosome, those alleles. So it's possible that if you want to genetically manipulate something, and you want to sort of manipulate the meiosis process and force certain chromosomes to be segregated in that process, there's not really any way you can get these three alleles separate, because they're all going to ride on the same chromosome. So they must all travel together. And so certain genes will be carried along. Certain traits go with other traits. And we touched on this a bit when we talked about breeding dogs and how we breed them for cute little faces and shapes and things, or behaviors. But along with that come the genes for serious health defects, riding along with it. So what we have to understand is that not all genes are independently sorted. Not all genes are independently sorted. That's not actually true. Certain genes may be linked with other genes because they ride on the same chromosome. So that's an important distinction that our new chromosomal theory of inheritance says instead of what Darwin had said. Or sorry, Darwin, Mendel. Okay? Now, here's some other interesting things. Mendel, basically I'm just comparing. Mendel had said that there is what we call the dominant allele and a recessive allele. These are the alleles, right? And what we've discovered is that that's not quite the case. It's not so black and white. We also now have something, what we call codominance. Codominance is when both alleles are expressed equally. So in other words, we get a condition where you can get a blending of traits. Both alleles are equally and fully expressed. So codominance means we get sort of variations like this. For instance, in cattle, here's an example of this. In cattle, you can get sort of a white cow. Here's a cow. Okay, here's white. And then you can get a brown cow. Oh, this cow has a big head and very little legs. There's the brown cow. Color them in brown. Expressed equally. For some reason, I can't get my brown color. Okay, here we go, brown. Thank you. Jumps around on me. So let's color rats. Crayon, brown. Good enough. Let's color this guy in brown. I'm going to color him in really well because I want him to look really, really brown. Now, if brown was dominant over white and they had babies, then the babies would have, we could do a Punnett square and figure out the percentages. If that was a homozygous dominant brown cow, all the babies would turn out brown, right? Hi. Okay, but here's what we find. What we find in certain cows, or in this particular situation, is that both alleles are sort of dominant. They are both expressed. So in codominance, what you get is a cow that is not white or black. It's not like it's one or the other. Excuse me. You get, oops, let's go back to black for a second. You get a cow. Here's our cow. And the cow is what we call roan. It's got black and white sort of all over the place. Patches of white, patches of black. So if I were to try to color this roan, it would be sort of dark area here, white area there, darky. Like not so much spotted, not like actually spotted. What you'd be getting is you'd be getting little white hairs everywhere and dark hairs sort of everywhere being expressed. So there would be all these sort of regions where the cow has, a mix of white. In your textbook on page 243, there's a reasonably good example of what we call a roan cow. And this means that both of the alleles are expressed. But they're not expressed in a way that mixes. They're basically, you're going to get sort of white or brown still. Okay? So each particular follicle of hair is growing, and it'll have in genetic instructions that will either end up being white or brown. And you'll get all this mottled sort of situation. Now there's another situation that can occur. And this is what we call incomplete dominance. So the other situation, incomplete dominance, and this is a situation in which neither allele completely conceals the other. And so what you get is an in-between phenotype. Okay? You'll notice that the above cow in codominance is still brown and white. It's not a shade of medium brown. Like it's actually brown and white because the brown and white is expressing itself both. But in this case, the in-between phenotype occurs. Let's say we have these two flowers, and we take a red flower. There's some nice red petals. There, a red flower. And then we will pick a white flower, which I'll have to just draw an outline because I can't take a same species of flower, which is white. Okay, I'm going to color that one in red. Crayon, red. Nice red flower. But actually, you know what I'm going to do? I'm going to take my pen again, because I don't like the way the crayon is portraying this. So let me make a thick red line. This has to be like nice and solid red. I don't want it to look like there's some red and white in it. It's solid. It's a red flower. So there we go. That looks better. So when we cross these two, what we end up getting is a situation where the red cannot completely mask the white, or vice versa, but instead of getting a flower that has red and white all through it, we get a mix of the two traits. And the flower that comes out in a pinky color. Well, let's see. Am I going to be able to find a color that's pink? Yep, right there. So what we would get then is, wow, okay. I thought I picked pink. Got to press OK, I guess. And then I got a dot. So what we get is a pink flower. And let me color that flower in that nice pink color. Wow. Wow, I am pressing things. I really am. Okay, here we go, pink. Okay, so it's important to see the difference. It's a slight difference, but it's an important difference. In the first case, both traits are expressed, right? And we get both of them occurring, like the roan cow with the white. Certain hairs are white, certain hairs are brown. It's all mixed. But in here, it's a blending of the two. So you get these intermediate blendings. And it often happens with colors of flowers and things. It's hard to really, I mean, there's lots of different examples, but this is the best sort of example. So we need a way of describing this. So we use a slightly different notation for this incomplete dominance. Because we can't use a capital and small because both of these are the same. So what we do here is for the red flower, we color and we put an R on the top like an exponent, a superscript. And this would be a white flower, like this. Okay? And then when we describe the genotypes, if you cross them, this one would be CR, CR, and this one would be CW, CW, right? So the gametes that are possible are just the CR, and here the only possible gamete is the CW. That's the only way to be white, right? Because if you had the mix, when you put these two together, you get a genotype of C with an R and C with a W. So this would be a red phenotype flower, and that's the only way to get red. This would be a white phenotype flower, and that's the only way you can get white. And then this, when the two are mixed, is a mix of the two traits producing a phenotype that is pink. So in this kind of crossing, it's all dominant, well, all capital letters, and we just keep track of the alleles with the little superscripts on top. And then there's only 3 possible situations. 2 of the same, or two that are different. And the two that are different would be the blending of the two traits. So that's incomplete dominance and codominance. Okay, I could give you an example. I didn't give you an example of codominance, though, did I? So I kind of went out of order. So this is an example of incomplete dominance. Here, I did. I give you the roan cattle. So that's kind of one example. But we didn't go into a cross or do any of the Punnett square things. So I'm going to take this. I'm going to go down. I'm going to go down below here. Whoops. Should have done this before. It's my fault. I'm going to do another example of the codominance. And this is a human example. Codominance. And it's something called sickle cell anemia. Sickle cell anemia. And this is a disease that is caused. It's caused by a genetic situation. I'm going to tell you how it works like this in a minute. So what happens in sickle cells is it affects your red blood cells. Normally, a red blood cell is round and it has sort of a little dent in the middle, right? Looks like a little donut with a dent instead of a hole. But in sickle cells, the red blood cell takes on a weird shape like this, like a sickle, like a curved shape. So this is what we call a sickle cell. And this is a normal cell. And of course, these are red blood cells, normal red blood cell. So this is a sickle red blood cell. It's important to remember that it occurs on the blood cells. Sickle red blood cell. Now, we need alleles, okay? And because this has something to do with the hemoglobin inside the cell, that's the protein that's being made, the hemoglobin is the actual sort of trait. And so what we do is we describe this using HB as our bottom letter, which stands for hemoglobin. And then we have the normal situation, which is considered to be an S. Oh, wait, no, that's a sickle. Sorry, normal wouldn't be an S. That doesn't make sense. Normal would be we'll call it A for normal. And then this one would be HB, and it would be S for sickle cell. Okay. So what happens, of course, is if you get the sickle cell trait, there's three different things that can happen to you. If you end up with the three possible genotypes that you could get from this, if you had this, that would result in a normal phenotype. All your blood cells would be normal. And if you had this, if you had both alleles, this would be sickle cell anemia. This would be a very, very dangerous disease. Because in the sickle cell form, the blood cells are not as good at carrying oxygen. So this means that you're going to have trouble getting oxygen to your cells, and it's a very serious condition. Okay? And it can even result in death. But what's interesting is if you get both alleles, the heterozygous situation, you get what's called, you're sort of a little bit of a both. You get sort of both traits. So what this means is you will have some cells that are sickle cells and some normal. Some normal, some sickle. So they carry the sickle cell trait, You carry the sickle cell trait right here, and it is expressed, but what's interesting is it doesn't seem to have a very big effect on your health. While some of your cells are sickled, there seems to be enough of the other cells that it doesn't really matter all that much. There are very slight, if any, symptoms of having this. So in a sense, you kind of live normally. But here's the really cool thing. The cool thing is that, so maybe we should write here, some normal, some sickle, but we'll put normal phenotype. We'll put normal under here. What's interesting is there's a disease. I'm going to write this disease in a different color, purple, malaria. And malaria is caused by a little protozoan, a little single-celled protozoan, very tiny. And what happens is it gets into the mosquito's saliva. And when the mosquito bites you, it goes into your bloodstream, and it swims around inside your bloodstream. And this little protozoan that's swimming around inside your bloodstream causes the disease of malaria. It turns out, though, that the sickle cell condition makes it harder for these guys to actually grow. So the sickle cell trait, the heterozygous, whoops, let me spell that right. Hang on, better start again. Monday morning. Heterozygous condition, the heterozygous carrier, right? So HbA/HbS, carrying both, has some immunity, I guess you could say, to malaria. So it's a weird thing where the sickle cells are not necessarily what you would want. And if you have both of the alleles, bad news. Sickle cell anemia, bad news. But if you have only one of the alleles, you're pretty much normal in terms of phenotype, but you do have some of those sickle cells which then interfere with the process of malaria infecting you. And so that condition makes you a little bit more, I said some immunity to malaria, or maybe we should say more resistant to malaria. It might be a better way of putting it. It's not really an immunity. That's not really a good word. More resistant to malaria. So what you find is that in places in the world where there is a lot of malaria, tropical regions, subtropical regions, Africa, Indonesia, places like that, even in the lower parts of Europe, like the Mediterranean Europe, malaria does exist there too. These individuals tend to have a slight advantage. And they can sort of withstand, or they can fight off the malaria a little more. And this is a case of a codominant situation. Because remember, if it was a dominant recessive situation, you could either have all round red blood cells or all sickles. There wouldn't be this mix. But the codominance allows for both. So that's another example of codominance. In fact, did I write that up here? Yeah. Codominance, and this is in humans, but it's weird in that it actually gives us an advantage against something else. So the thing with genetics is there's no good or bad. Every genetic change could possibly confer a benefit or a negative effect. And it depends on the environment you live in. And that's, of course, the whole mechanism of evolution, which we're going to learn later. Okay, one more thing to talk about, and that is the idea of multiple alleles. Multiple alleles. So far, we've seen that there are two forms of the alleles, and you have two of them. But it's possible to have more than two. So what this means is more than two alleles. Now, in your genes, you can only have two, but there might be 3 or 4 forms of the allele. Right? You can only have two because you only have 2 homologous chromosomes for every gene. And so you can only get 2 of the alleles, but that doesn't mean that there's not more possible alleles out there. And the best way, or the best example of multiple alleles is the human blood groups, or at least it's a commonly known example. They follow a multiple allele situation. Human blood groups. Okay, so here are the alleles. Here's what you can get. They use an I as the base letter. And I with a capital A. Now again, this is not a case of incomplete or, well, yes, it kind of is and it isn't. So let's just go. Here's the allele for what we call the A antigen. So basically, these alleles produce a particular kind of protein on the outside of your blood cells. That's what the antigen is. So it produces an antigen. Antigen A, or A antigen. So remember, an antigen is a special protein on the outside of blood cells that serves as a way to identify and whatnot. Okay, then there's allele B, which is this, which produces the B antigen. And then there is another allele, which is the small I. Now, the reason we're using a capital I for the first 2 is because they are dominant over the small I allele. The small I allele produces no antigens. Okay? But if you have the A and the B together, they are codominant with each other. So you have a case here where there's 3 different possible alleles, and you can have any 2 of them in your genes. But there's 3 options. And depending on what you have, it could be a case of dominant recessive. The little baby I is recessive to both of the other 2 genes, but the other 2 genes are codominant with each other. So you get these interesting situations where if you have this genotype, where you have both of the A alleles, then you have A antigens, and we call that blood type A, right? Type A. But if you were to get one of the A and one of the B, that's a codominant situation. If you were to get one A and one B allele, depending on your genetic makeup, then you would be making, well, actually, why don't we do both B's first? Let's do both B's first. So, B, this would make B antigens. And you would be type B. Okay? Now, if you were to get these 2, that would be no antigens, and you would be type O. Now, what's interesting is the combinations when you mix them. So if you mix them, you can have IA with a little I. The A antigen allele is dominant to the recessive little I, and so this would still produce A antigens. So you are still a type A. You're still, in other words, the little I is masked, right? And if you combine a little I with the B allele, you are still going to produce B antigens because you are masking that little I allele, and you will still be type B. The only other combination is if you get one of the capital A ones here, the dominant, and this is the codominant situation, where you will produce both A and B antigens. That's supposed to say codominant. Codominant. Codominant situation. Both A and B antigens. will be produced. Wow. For some reason, I can't spell or do anything that I need to do today. There we go. Both. And you would be type AB. So, when you look at the phenotypes, there are two possible phenotypes for type A blood. Because it's dominant, those two possible genotypes produce the one phenotype of type A. And likewise, these are the two possible phenotypes or genotypes that you could have for type B blood. These produce the same result, type B. In other words, your blood cells will have the A or the B antigen, depending, in both of these cases. Then you can have the combination of little I, which produces type O, no antigens. And you can also have the codominant situation of one of each of the dominants, which produces type AB blood. Multiple alleles. So we could do a little cross just to have fun with this. We could imagine some parents. So let's do an example of a cross, right? Let's say you have a mother who has type A blood, but she is heterozygous, right? Heterozygous. And then you have a father who has type B blood, and he's also heterozygous. What that means is we can determine their blood types, or their genotypes. Type A blood has to have the allele for type A, but if it's heterozygous, it has to have a different allele. And the only way you can have type A blood with a different allele is if you have the recessive version. If you had the IB, your type would be AB. It would be codominant, right? So that's the only way you can have type A blood and be heterozygous. And the same is true for the father. The father can only have this possible genotype because he has to be type B, but he has to be heterozygous. So then if we did a cross to see what would happen, we could look at the possible gametes. Well, the mother's gametes could either be, let me do this in black. The mother's gametes, we'll put along the top, are either IA, I can do them in the same color maybe, just so you can see where I'm going with this. So the mother was type A, right? So there's one possible genotype. The other, or one possible allele, the other allele would be this. And the father, who was type B, could have either this one or this one. Those are the possible gametes, right, when you separate those genotypes. And so then we can figure out the possible combinations. Well, this would be IAIB. This would be IB with a little I. This would be IA with a little I. And the last one would be this little I combination. So if we look at the possibilities for type A blood, there's only one genotype here that would give us type A blood. So we could write type A is 1 out of 4, right? 1 out of the 4 possibilities. Type B, there's only one genotype that could give us type B blood, and it's this one. So that's 1 out of 4. Then there's another possibility of getting type O, which is the possibility of this one. So that's 1 out of 4. And then the other possibility is codominant situation, which would be type AB, which is also 1 out of 4. So in this cross, we can get an equal sort of chance of all the possible blood type combinations. Now, what happens, of course, if one of the parents had been homozygous, if one of the parents was a homozygous situation, like, say, parent A here, let's go up here. Whoops, where are we? Right here. If instead this had been homozygous, and this genotype was this, that's the homozygous type A blood type, then if you went down to the square, you could see how that would change a lot of our situation. This would have to become this situation, and this one would turn into this situation. And so it would be impossible for this particular cross to produce type O blood. It just would never happen because there's no combination that would result in the two little eyes, right? It also means that there's a 50-50 chance of producing this one. This one, it was 1 in 4. I guess I didn't write that. It was 1 in 4, but now it's 2 out of 4. So there's a very good chance of that. And then there's a 2 in 4 chance of producing type A blood here and here in the heterozygous condition. So you will never be able to get any type Bs from this, and you will never be able to get any type Os. And so now we have a cross where there's only 2 possible outcomes. So depending on the parents' genotypes, the children will have various percentages or probabilities, which we can figure out in Punnett squares, just like this. But because there's 3 different alleles, it's a little more difficult, and this involves, of course, codominance as well as the dominant recessive all kind of mixed together. So it's a little bit trickier, but it can be done. So that's multiple alleles, and I think that's as far as we'll go for today on this video. And then we'll, one more thing. I wonder if we should, yes, we should. One more thing that's different from what Mendel understood, and that is what we call polygenic inheritance. This is a situation where there's more than one gene involved in a phenotype. And this is actually quite common. And a good example of this is skin color. So we had talked about the production of melanin, a dark pigment that contributes to darker skins. But there are multiple genes involved, and so it's difficult to actually figure out what skin colors can do. So watch this. I'm going to try to give you a chart here. For skin color, there's going to be 3 genes involved. Right? We're talking about skin colors here. Skin color. 3 genes involved. Let me get to a thinner pen here. And so the genes are, there's going to be one where you can have these alleles, A, you can be heterozygous, or you can be homozygous recessive for that gene. Then there's another gene and the possible things you could be are homozygous dominant, heterozygous, or homozygous recessive. Those are the 3 ways that gene could work out, right? And then there's a third gene. So these are the 3 ways. I'm going to put a little C here with a little, I'll do it like a scripted C so it looks different. Okay, those are the 3 genes, and those are the 3 different possible genotypes that someone could have. So far, so good? All right. So what does it look like when you get all the different combinations? If you start looking at the combinations, you could have, this AA and this BB and this CC. Well, what would that look like? And how would that be different from having this combination, and this combination? And there's lots of combinations, right? Because these combinations, you've got 3 times 3 times 3 different possibilities. So there's lots. So what we can do is we can sort of draw ourselves a little chart. Okay, so I'm going to put a little, black. Eraser. I'm going to put a little black line along the bottom. Okay? And I'm going to divide this into a bunch of sections. Am I going to be able to fit it all in? That's the problem. I will try. So I'm going to put a middle section here. And then on either side, I need another 1, 2, 3 sections. 1, 2, 3 sections. Okay. The darkest possible skin occurs when you have homozygous dominant of all possible genes, all expressing. Okay? So in that case, you would have the darkest possible skin, and it would have a genotype of both capital A's, both capital B's, and both capital C's. So there's your 3 genes, and you have the dominant situation, the homozygous dominant of all. Now, the next one over would be the situation where only one of these combinations has a recessive allele. And so that could be the following 3 situations. It could be 2 capital A's, 2 capital B's, but 1 little recessive C. Or it could be the recessive allele could show up in the B gene, capital, capital. Or it could show up in the A gene. So, in other words, one of your alleles on one of those 3 genes is a recessive allele. So your skin is going to be slightly lighter. And then in the next section, in this section you're going to have 2 recessive alleles. Just 2. Now there's many combinations of that. A, and then we'll put B, little b, big C, little c. Now you have 2 recessive alleles in the B and the C gene. Or maybe it's A, oops, sorry, maybe I should do the little a here. Hang on. I'm trying to squeeze it all in, so it's kind of tricky. A, little a. We could have 2 capital B's, and then a C and a little c. Or we could have 2 capital C's. So we could have an A with a little a B with a little b, and 2 capital C's. So you see, in all those 3 situations, there's only there's 2 recessive alleles. But you could also have 2 recessive alleles on 1 gene. So you could also have a situation where you got all capital A's and B's, but 2 baby C's. Or you could have 2 baby B's, right? So you can see how there's far more options here for getting 2 recessive alleles. Or you could have 2 baby A's, 2 recessive A's. Those are all the ways you can have 2 recessive alleles. Then guess what? As we move into the middle, we would see 3 recessive alleles. So, A and a little a. 2 capital B's and 2 little c's. There's 3 recessive alleles. Or A, little a, 2 capital B's, and 2 small, 1 big C, 1 little c. We want 3 recessive alleles here. Then, capital A, B, little b, and 2 little c's. There's 3 recessive alleles. Then we could get capital A and a small a, 2 little b's, 2 big c's. Then we could get 2 big a's, and then we could have 2 little b's, and then 1 big C, 1 little c. Or we could have 2 little a's, and we could have a big B and a little b, and 2 big C's. Or we could have a capital A, a small a, a big B, a little b, and a big C and a little c. So there should be 7, I think, different ways to get 3 recessive alleles in combinations. Okay? Then we get to 4. So the numbers start to go down again. Here's one way to get 4 recessive alleles. Here's another way. And yet, here's another way. Another way, A, B, C, and little c. And another way would be A, 2 little b's, and a big C, little c. And the last way would be an AA, BB, little c, like that. Now I wrote a little bigger, but you'll notice that 1, 2, 3, 4, 5, 6, there are 6 combinations. So we'll go back to the combinations later. And then over here we need to have 4. So that was, wait a minute. We need 5 now, 5 recessive alleles. So AA, B, big C, little c. A, big B, little b, 2 small c's, and a capital A. Those are the only ways you can get 5 recessive alleles. And then there's only one way to get all 6 recessive alleles, which aren't expressed, remember? So if you have a dominant situation, or a situation where you're going to have, the more recessive alleles you have, in other words, the less and less and less this gene will be expressed, or all 3 genes. So if we look at the numbers, if I'd have drawn it all, it would have made a nice little sort of shape like this, but my third column here, I made my letters too big. 1, look at the numbers. 1 combination, 3 combinations, 6 combinations, right? 1, 2, 3, 4, 5, 6, 7 combinations, and then down to 6 again, and only 3, and only 1. So what that means is that we could think about the height of the column as the frequency. The frequency, how many different ways there are to get these combinations, means that's in a population, that's where you'll have the most phenotypes. Most people will be in the middle. Now, what happens, of course, is these are skin tones. I can't shade them because they don't have enough colors, but this would be the darkest skin tone, and this would be the lightest skin tone. So if you were from the heart of Africa, living in a sub-Saharan African continent, like country, you would probably have very dark skin. And you would have either condition 1 or maybe condition 2 here. You'd either be in this situation or this situation, darkest of possible skin tones. If you're like me and you're a little bit more European in descent, where skin tones are lighter, you're either in the 1 or the 2 section, right? And if you're from a Mediterranean country, let's say, you might be somewhere in the middle up here, where you have a skin tone that's sort of not totally white, but not totally dark either. If you are of Asian descent, you'll probably be somewhere in the middle here. Inuit descent, Native North American, you can look at all the different tones of skins and ranges of colors, and really they all come down to degrees of melanin. And you can see that in a population, though, if you just let everybody breed, if the whole world just starts randomly breeding, this is going to become the most common phenotype, a blend in the middle. This has only started happening recently because in the past, people tended to breed within their own little small groups and countries because we couldn't travel very far and groups of different races weren't easily mixed. But in today's world, people mix all the time. There are people of different descents all over the world. So it's very easy to have people with very light skin mating or mixing with people with very dark skin and all in between. And if that continues, what we can expect to happen is we can expect to see more and more and more people falling in that middle range because it's the highest possible situation. The most possible gene combinations will get you there. So it's kind of like everybody's just going to blend to something in the middle, which kind of makes sense. So here we have 3 different genes controlling skin color. 3 different genes, and so you can see how it gets to be quite complicated. That's called polygenic. I keep saying Darwin. Mendel really didn't have a handle on this. He didn't understand that this was a thing. But we've now figured it out because we have the chromosomal theory, which allows us to place those alleles specifically on a chromosome in a certain spot, and then we can deal with all kinds of things that Mendel couldn't explain. And that's as far as we'll go today.